Okay, everybody, this is the second part of the video lecture for Chapter 13 on meiosis and sexual life cycles. Remember at the end of the first part of this, we had wrapped up by talking about life cycles. I gave you the example of human life cycles specifically. Talked about, in particular, how life cycles of most sexually reproducing organisms are actually very similar, especially between all mammals, for example. And the key point about life cycles, sexual life cycles, is that they alternate between a diploid, typically multicellular stage, and a haploid, single cellular stage. And the transitions from diploid to haploid and from haploid to diploid are meiosis and fertilization, respectively. Okay, so in the second half of this chapter, we're going to dive into the mechanics of what's actually going on in meiosis. Now remember, meiosis is a special type of cell division that produces gametes. So gametes are sperm and egg, okay? And gametes are unique compared to the rest of our somatic cells. Remember, somatic cells are body cells. They're skin, muscle, nerve, bone, blood, for example, those types of cells. They play no role in whole body reproduction or whole organism reproduction. But that's not true of gametes, sperm and egg. Sperm and egg, for one, are all genetically distinct. They're all genetically different from one another. That's not true with our somatic cells that are produced through mitosis. All of those are identical with the exception of the rare and occasional mutation. All right. So gametes are genetically distinct because of some unique events that occur in meiosis that we'll talk about. Secondly, gametes have only half the normal number of chromosomes, all right? So instead of gametes being diploid, like the rest of your somatic cells are, gametes are actually haploid. So in humans, a human egg would have 23 chromosomes, a human sperm, 23 chromosomes, a human skin cell, 46 chromosomes. That's the difference between the skin cell, which is diploid, 2n equals 46, and the sperm and egg, which by themselves or individually are haploid, n equals 23. The reason why we have sperm and egg being haploid is that because when they fuse during fertilization, that takes us back to a fertilized egg, a zygote, which has the diploid number. So because meiosis cuts chromosome number in half as it produces these two types of gametes, it's often referred to as reduction division, okay? And that keeps chromosome number constant generation after generation. Otherwise, you get a doubling of chromosome number if you got a diploid egg fertilized by a diploid sperm, for example. Every generation, chromosome number would double, and embryos wouldn't survive that. Okay, so what we're talking about next then is, again, meiosis. And in meiosis, we have two separate divisions. We call them meiosis 1 and meiosis 2, all right? Now, all of the things that make meiosis unique and distinct from mitosis occur in meiosis 1, all right? Meiosis 2 is essentially just mitosis. It's the same basic thing. You've already seen it in the last chapter. So we're going to talk about that a little bit, but for the most part, we're going to focus on the unique aspects of meiosis 1 that result in the outcome, the cells that are produced as a result of meiosis being so dramatically different from the cells that are produced through mitosis. All right. Now, we're going to start diving in next to the stages of meiosis, okay? So if you think about the cell cycle, we still have the same cell cycle. We have interphase, which may be 90 or 95% of the overall life of the cell, all right? And that's broken up into G1, S, and G2 phases, just like we saw back in the chapter on mitosis, all right? But again, all the unique things that are happening here are in the meiotic phase. So now we call it the meiotic phase. This is the part of the cell cycle where the gametes are produced, okay? All right, and one thing I'll say here is that because chromosome number changes as we go throughout this process, remember, meiosis cuts chromosome number in half. Students very often find certain aspects of meiosis confusing, okay, especially keeping track of chromosome number. So here's how you do it. If you're ever confused about how many chromosomes you're dealing with in a particular stage of meiosis, you always count the centromeres, all right? So let's take a look at that. Let's say we have a chromosome that is not replicated. So I'm drawing a chromosome here as it would appear, let's say, before S phase, right? That's a non-replicated chromosome. Remember, you have the centromere right there. I've drawn the arrow to it. Now, after S phase, so in S phase, we have DNA replication. After S phase, that chromosome then would look like that. So now that is a replicated chromosome, all right? A replicated chromosome. Oops, sorry about that. So that's a replicated chromosome, has two identical sister chromatids, one here on the right and one on the left. Those are referred to as sister chromatids. They are duplicate copies of the same chromosome. All right, so a lot of times students will look at this single chromosome here and they'll say, okay, well, that's one chromosome. Yes, it's a non-replicated chromosome. But then they'll look at the replicated one you get after S phase and they'll say, okay, that's two chromosomes. No, it's not. It is one duplicated chromosome. It consists of two sister chromatids, but they're connected at a common centromere. So the way you count chromosomes here, no matter what stage of meiosis we're talking about, is that you count the number of centromeres. Here we have one centromere, so one chromosome. Here we have one centromere, again, one chromosome. What's the difference? Non-replicated chromosome, replicated chromosome. They're both one chromosome, all right? This one, however, because it's replicated, consists of two sister chromatids, all right? So what's going to happen here is that chromosomes will duplicate during interphase, S phase in particular of interphase, and our resulting sister chromatids will be closely associated along their length. They're lined up with each other by being attached to that same or that common centromere, all right? Now, keep in mind, we have two copies of every chromosome, correct? So let's say that this replicated chromosome is the one that you got from your mother. You will have another one that you got from your father, all right? And those will have the same sets of chromosomes on them. I'm sorry, the same sets of genes on them. So if you have chromosome number 4, let's say from your mother, and number 4 from your father, they would be identical in the sense they would contain the same sets of genes. If you read the list of genes from top to bottom, you would see they're the same on this chromatid, the same on that chromatid, the same on the father's two chromatids as well. So, because these two chromosomes have the same sets of genes on them, we call them homologous chromosomes. They have the same genes, all right? They're also sometimes referred to as homologs, okay? Homologs. All right. So, in humans, we have 23 pairs of chromosomes. This is pair number 4, for example. You'd have a pair 1, 2, and 3, and a pair 5 through 23 all the way down the line, all right? Each homologous pair after DNA replication consists of 1, 2, 3, 4 sister chromatids. Ultimately, what's going to happen here in these two separate cell divisions, meiosis 1 and 2, is that these 4 chromatids will be separated into 4 separate haploid cells. Those cells become our gametes. All right? Okay, so let's take a look. This is essentially what's going on. So let's imagine we have a simple case here where we have one pair of chromosomes. Just one pair of chromosomes. So you're seeing a cell here prior to S phase. So what they typically do is color code these. So let's say the blue is the father's chromosome, the red is the mother's. Okay, what do I mean when I say that? I mean your father and your mother, right? We get our DNA from our parents, we use it for a little while, we then pass it on to the next generation when we reproduce. All right, that means half of our DNA came from our father, half of it came from our mother in the form of these chromosomes. All right, prior to S phase, we have 2 non-replicated chromosomes, one centromere there, one there. After S phase, we have 2 replicated chromosomes, one centromere, again, just 2. But now we have 1, 2, 3, 4 sister chromatids. All right, so this pair of homologous chromosomes lines up gene by gene. Something will happen there that we'll see later on. We won't get to it just yet. But essentially, meiosis 1 is going to take and separate those two into two separate cells. So we start off with a diploid cell, 2 copies of each chromosome. One of them goes into this cell, one into that one. So now we have 2 haploid cells. Diploid. In this case, we have 2 centromeres. Haploid. 1 centromere in that cell, 1 in that one. So meiosis 1 then has cut chromosome number in half. All right. Meiosis 2, that you see down here, then takes the chromatids and separates them by splitting that centromere and puts each of them into separate cells. Right? So we end up with 1, 2, 3, 4 daughter cells, as they're called, that are all haploid, and they have the potential then to go on and become sperm or egg. All right. Now, it turns out in sperm production, all 4 of these would become sperm. In egg production, only one of them becomes the egg. The other 3 become what are called polar bodies. And basically, they're just garbage cans for excess chromosomes. A human egg is about 100 or 1,000 times larger by volume, I should say, than a typical human sperm. And that's because it needs all that cytoplasm, which is essentially the yolk of that egg that contains all the nutrients, all the morphological determinants called proteins and things that will kickstart the process of embryological development if that egg is fertilized. The sperm brings one thing to the party, and that's it. It brings DNA from the father. It's like a little missile containing DNA. It fertilizes the egg, the 2 nuclei of those cells fuse, and then the egg, which has all that cytoplasm, the yolk, all the nutrients and things, takes over and begins the process of embryological development. That's why it's so much larger than the sperm. It has to have all that yolk. So essentially what happens during egg production is that these cells, if these 3 cells are the ones that don't become the egg, their cytoplasm is concentrated into the one cell that does become the egg, and these just get rid of excess chromosomes. They're called polar bodies. Point is, you get 1 egg produced for the 4 potential daughter cells at the end of meiosis in egg production, whereas in sperm, all 4 of those become sperm. In sperm production, all 4 of those become sperm. All right, so if you remember back to mitosis, we had 4 basic subdivisions or subphases of mitosis. We had prophase, metaphase, anaphase, telophase, and then also cytokinesis. We have the same things in meiosis, except we call them prophase 1, metaphase 1, anaphase 1, telophase 1. And then for the second division, which is meiosis 2, we call them prophase 2, metaphase 2, anaphase 2, and telophase 2. All right? But again, all the things that make meiosis unique and different from mitosis occur in meiosis 1. All right? Okay, so let's look and see what's going on. Okay, so these are the 4 subphases of meiosis. All right, meiosis 1 rather. Okay, so we have prophase 1. Okay, and in prophase, the same basic things happen in meiosis that happen in mitosis. You have the chromatin that condenses into chromosomes. You have your nuclear membrane that's breaking down, disintegrating because it's in the way. You have one centrosome, that migrates to the other pole of the cell, and we have the spindle microtubule fibers forming between them, building that spindle apparatus, which again is the machine that's going to sort of arrange the chromosomes. All right, but now look at the chromosomes. We have a process whereby each chromosome from the mother finds its homologous partner amongst the father's chromosomes. So number 1 from the mother finds number 1 from the father. Number 2 from the mother finds number 2 from the father, and so on. And they line up, just as you see right here. They line up just like this. They line up gene by gene, in other words. So if this is the father's chromosome, that's the mother's, all right? And that process of them lining up is called synapsis. So synapsis is when homologous chromosomes find each other. All right, let me zoom in a little bit. Now notice right here, we have what's called chiasmata. This is a process called crossing over, all right? Crossing over is when you have the two innermost chromatids of that homologous pair, say, for example, this one on the inside of the fathers, this one on the inside of the mothers, they actually lay across each other. And they, the points at of crossing over are called chiasmata, and the sections of the chromatids beyond that. So let me show you again right here. So this section, for example, on the fathers and this section on the mothers trade places. They swap positions. So what you end up with is part of the mother's chromosome now on the father's chromosome and part of the father's on the mother's. And this happens at each of these crossover points. Typically there are 2, sometimes 3 crossover points, 2 or 3 chiasmata, okay? That's a really key process in meiosis. That is why the cells that you get at the end of this are genetically distinct from one another compared to what you get in mitosis. This doesn't happen in mitosis, okay? This is a process of taking your parents' DNA and jumbling it up as we make our gametes, all right? It's why we get, one of the reasons why we get so much genetic diversity in the next generation. All right, all that happens in prophase 1. Now, metaphase 1. Now you see that homologous partners line up in essentially double file, right? So you have this one, which is the fathers, that one, which is the mothers. They line up side by side at metaphase 1, okay? Metaphase 1. The fact that they line up in double file like that is why we get the reduction division, because notice that this microtubule fiber is only connected to a single side of that blue chromosome, that father's chromosome. This one is connected only to a single side of the mother's chromosome. In mitosis, you have microtubule fibers connecting to both sides of each chromosome. And when they retract, they split the centromere and the chromatids go their separate ways and become full-fledged chromosomes. Well, in this case, what happens is that because they line up in double file, when the fibers retract, as you see here in anaphase 1, it's entire chromosomes that get pulled away from each other. Let me zoom in. All right. And so you can see there's an entire chromosome here, a replicated chromosome being pulled that way. Same thing that way. Same thing for the others as well. But notice now they're a mixture of the father's and the mother's DNA. This one is mostly paternal DNA, the father's, with some maternal mother DNA on it. This one's mostly maternal DNA with some paternal DNA on it. Notice also there's one chromatid, this one, and that one, the ones that did not participate in crossing over, that remain 100% the original parent's DNA. So what crossing over does is it produces chromatids that end up being a mixture of both parents' DNA. We call those chromatids then recombinant because they're now a mixture of DNA from 2 distinct individuals, 2 distinct sources, okay? The ones that do not participate in crossing over end up, or are rather called, non-recombinant, all right? Okay. Also, notice again, back in metaphase 1, we still have our non-kinetochore microtubules, those that don't connect to kinetochores of centromeres, that help to sort of, again, elongate the cell and push the poles apart, which during anaphase, again, helps to separate those chromosomes from each other. Okay, same thing we saw in mitosis. All right. Anaphase 1 technically is the reduction division step. That is cutting chromosome number in half. We had 1, 2, 3, 4, 5, 6, count your centromeres here, diploid number. Now we have 1, 2, 3 moving to what will become that cell, and 1, 2, 3 moving to this pole, which will become the other cell. Telophase and cytokinesis, we have a new nuclear membrane forming now around each distinct set of chromosomes. Cytokinesis begins with the cleavage furrow in the same way we saw it in animal cells in mitosis. All right. Now, if we zoom in a little bit, take a look. We have in this cell, in this nucleus rather, 1, 2, 3 chromosomes. In this nucleus, 1, 2, 3. We started off with one nucleus that had 1, 2, 3, 4, 5, 6. We've gone from a diploid number of 6 to 2 haploid nuclei with a haploid number of 3 each. And that's because of anaphase and the fact that they line up in double file at metaphase 1. All right, so now we have nuclei and ultimately cells that are haploid, but also notice they're now a mixture of both parents' DNA, a mixture of blue and red. The chromosomes are, and that tells you they're now genetically different from each other and genetically different from the cell that they came from. Okay. Now, meiosis 2, as I said, we have prophase 2, metaphase 2, anaphase 2, telophase 2, and also cytokinesis. And essentially what's going on here is what we've already seen in chapter 12, mitosis, all right? So these two cells that we end up with at the end of meiosis 1 will each then split to make 2 more cells. So we'll get 4 from those 2, and each of those 4 will be genetically identical and have only half the amount of DNA or chromosomes that the original parent cell here started with. All right, let's take a look at that. So imagine these are the 2 cells we ended up with at the end of meiosis 1, okay? So almost as soon as those nuclei form, those nuclei break down again, nuclear membrane breaks down, each one of them. We have a new spindle apparatus forming in both of them, okay? And the microtubule fibers will connect to, in this case, both sides of each centromere, just like they do in mitosis. So now here we are in metaphase 2. Now you see chromosomes are lined up in single file in both cells, not in double file. There's no longer any homologous chromosomes finding each other through synapses and lining up left-right. That's done with. Now you see that the chromosomes line up in single file. You can see they're a mixture of paternal and maternal DNA, right? And then in anaphase 2, it's the spindle fibers from each side of the centromere retracting, splitting the centromere, and separating that chromosome into 2 sister chromatids that are now chromosomes in their own right. All right, so we'll get these 3 going into one cell, these 3 into a second, these 3 into a third, and these 3 into a fourth. All right, cytokinesis happens at the end of telophase here, and we end up with 1, 2, 3, 4 haploid daughter cells. And if you look at that distribution in blue and red, you'll see that all 4 of those cells are genetically different from each other because of the crossing over that occurred back in meiosis 1. All right, so, let's put all this together then. Okay, sorry, this is kind of small, but that's why I showed the 2 figures first. So here we have meiosis 1, all right? So again, unique events. We have the homologous chromosomes finding each other during synapsis, as you see there, there, and there. We have the crossing over, again, which is when they actually exchange fragments, and that mixes up your parent's DNA. That's in prophase 1. Metaphase 1, chromosomes line up in double file, as you see here now, okay? And then anaphase 1, it's entire chromosomes that separate from each other, and that's why you get the reduction in chromosome number, okay? All right, so at the end of this then, in meiosis 1, we end up with 2 cells, each that has half the number of chromosomes, 3 in this case, 3 here, compared to the cell they started with, which had 2, 4, 6. All right, also, these 2 cells are genetically distinct from each other. They then go on and enter meiosis 2, which is basically mitosis, where all the normal things we think of occurring in mitosis take place. There is not another round of DNA replication here that would defeat the purpose of reduction division here. So basically this is these 2 cells going through mitosis that we technically call meiosis 2. It gives us 4 haploid, genetically unique daughter cells. Again, in sperm production, all 4 of those become sperm. In egg production, maybe that one becomes the egg. These other 3 become polar bodies that just help to concentrate the cytoplasm in the egg, and that are a way to take off or get rid of those extra chromosomes that didn't go into the egg. All right. So let's do a comparison then now between mitosis and meiosis. Okay, there are a lot of similarities. For example, you have the spindle apparatus basically doing what it does, the same thing in both of them. What's different is the way the chromosomes are arranged. The crossing over that you get because of the synapsis of homologous chromosomes in meiosis 1, and the reduction division that you get because the chromosomes line up in double file meiosis 1, metaphase 1 and meiosis 1, that does not happen in mitosis. Okay, so mitosis conserves number of chromosome sets. It does not change number of chromosomes. So it produces cells that are genetically identical to the parent cell. There's no crossing over, nothing like that. All right, all of your skin cells are clones of each other and clones of the cells they came from, unless a random mutation happens to make one a little bit different. In meiosis, that reduces number of chromosome sets from 2, what we call diploid, to 1, what we call haploid, producing cells that differ genetically from each other and from the parent cell that they came from. All right, let's look at this. So let's imagine we take a parent cell, diploid number of 6, and run it through mitosis first, then meiosis second. All right, so let's say this is in G1 of interphase. S phase is where we have chromosome duplication, and now we have each chromosome duplicated. Each one consists of 2 sister chromatids. So do the accounting. We have 1, 2, 3, 4, 5, 6 chromosomes there. They're unreplicated. After S phase, they're all replicated. Again, 1, 2, 3, 4, 5, 6. Yes, you do have 12 chromatids there, but because they're attached to the same centromere in each chromosome, you still only have 6 chromosomes. Just now they're duplicated, right? Okay. In mitosis, they line up in single file, spindle fiber, microtubule attached to each side. Fibers retract and distribute those chromatids rather, to separate cells. Now we end up with 2 identical diploid cells. 1, 2, 3, 4, 5, 6. 6 over here, 6 in the original parent cell. No reduction in chromosome number. And no crossing over, so they're all identical. Now take this cell, run it through meiosis. So S phase converts non-replicated chromosomes into 6 replicated chromosomes. Count centromeres. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Now you see they're replicated, but now we have synapsis occurring. Homologs find each other, and they line up gene by gene. It's not like that's chromosome 1 from your father, number 7 from your mother. That's 1 and 1, 2 and 2, 3 and 3, and so on. All right, crossing over occurs. They line up in metaphase 1 in double file. Compare that to single file in mitosis. And again, because you have a fiber attached to only one side, that side, and one side here, when those fibers retract in anaphase, this entire chromosome goes that way, this entire chromosome goes that way. That's what reduces our 6 to 3, 6 to 3. And the crossing over now has converted them into recombinant chromosomes. Look at the distribution of paternal and maternal DNA, blue and red, and you see the pattern is different in each of them. So these 2 cells are now haploid, 3 chromosomes, 3 versus the 6 we started with. They're also a mixture now, a recombinant of the 2 parents' DNA. Take each of these cells, run them through mitosis, and what do you get? 4. 1, 2, 3, 4 haploid daughter cells that now you can see, because of the distribution of blue and red, are all genetically different from each other. All right, so to summarize then, there are 3 events that are unique to meiosis, and all 3 happen in meiosis 1, as I've said. We have synapsis. That's where homologous chromosomes find each other, your father's and your mother's, and crossing over. That happens all in prophase 1, all right? At metaphase, you have homologous pairs lining up in double file at the metaphase plate, that imaginary plane in the center of the cell that we talked about in the mitosis chapter, right? And then third, you have separation of homologs in anaphase. All right, synapsis and crossing over is point number 1. Homologous pairs in the metaphase plate of meiosis 1 is number 2. And separation of homologs during anaphase 1 is number 3. These are the 3 events that are unique to meiosis and that make the end result of it so much different than what you get from mitosis. All right. So now we've gone through the mechanics of what's going on in meiosis. Now let's talk about genetic variability. So what meiosis is going to do is it's going to take existing genetic variability that's in our parents' chromosomes, and it's going to amplify it. All right. So how do we have existing genetic variability? Well, the only way that you get new versions of genes, different versions of an original gene, let's say, is through a past mutation. A mutation is just a typo that the enzyme that copies DNA makes. It's an error that it makes when it's copying chromosomes. We'll see how that happens in chapter 16. All right. Now, these mutations are the ultimate source of all genetic variability because they can produce new versions of genes, right? So you may have a gene for eye color, let's say, on your mother's chromosome number 7, let's say. Your father's chromosome will also have that same gene, but it might be a different version. Maybe the one on your father's chromosome codes for brown eyes. Maybe the one on your mother's chromosome codes for green. They're the same gene. They code for eye color, but there are different versions of them because of a new mutation in the past maybe has now produced a green version that makes a green eye or codes for green eyes. All right? Okay. So mutation then produces new versions of genes, and we call those versions of genes alleles. Okay? These are just different versions of genes. Mutation produces alleles. What meiosis does is it reshuffles the alleles and can bring them back together in the next generation in a wide variety of different combinations, many of which may have never existed in the history of the species. All right? Okay. So we have that original genetic variation, alleles, present in our parents' chromosomes and our cells. And that's a result from past mutation, as I've said. What meiosis and fertilization is going to do is basically amplify that variability and bring it back together in the next generation in new combinations. All right. So we basically have 3 mechanisms that are going to contribute here to genetic variation. I'm going to write them down. We have what's called independent assortment chromosomes. All right. We have what's called crossing over, and I've already talked about that actually. Okay, that's when our parents' chromatids exchange fragments. And then we have, oops, I'm sorry. Go back to that. My mistake. And then we have what's called, changed colors on me, random fertilization. Okay, a random sperm fertilizes a random egg, and that's what random fertilization is all about. Okay, let's talk about independent assortment first. So in independent assortment, this has to do with the way that the chromosomes, the 2 chromosomes of a homologous pair line up. So let's say we're talking about chromosome pair number 1 in humans, all right? So let's say that for this pair, the mother's is on the left and the father's is on the right, okay? Pair number 2, it might well be that the father's is on the left and the mother's is on the right. And then pair number 3, it could be the other way around again. It could be this way or the other way. It's only 2 ways they can line up, right? The point is that each of these pairs, this one, and this one, line up irrespective or independently from how the other pairs line up. That means there's nothing saying that because this pair lined up this way, mother's on the left, father's on the right, that this one has to line up the same way or the other way. Each one is the flip of a coin, basically. All right? So that means that in humans, where you have 23 pairs of chromosomes, there are actually 8.4 million possible lineup combinations. Imagine it this way. Pair number 1 lines up this way with the mothers on the left. And then pairs 2 through 23 line up with the fathers on the right. That's one combination, isn't it? Second combination. Mothers on the left, pair 1. Pair 2, mothers on the left. All the rest of them, fathers on the right. That's another iteration, isn't it? So there are 8.4 million different ways or combinations like that in humans in terms of how those chromosomes can line up. That represents a lot of possible genetic variation there. That's independent assortment. We're going to see that again in the next chapter on genetics. Now, while these are lining up, don't forget, We have crossing over between those two innermost chromatids. That's jumbling up our parents' DNA. Crossover events occur on average 2 to 3 times per chromosome pair. How many times they occur is random. Where they occur along the length of that chromosome pair is completely random, cannot be predicted. That throws in an enormous amount of genetic variation, as we'll see later on. Then you take one random sperm, and that sperm is probably one of a million or more in a single male ejaculate, and you use that to fertilize a random egg, one of potentially 200,000 released every month during a human female's reproductive years. And which of those eggs and sperm are involved in that is completely random. You run the tape of time back to the magic night none of us want to consider, the night of our conception, and a different sperm fertilizes a different egg. And instead of you getting you get somebody as different from you as maybe your siblings are from you. Okay? Each of us is a complete crapshoot in terms of how we turn out. We are a result of a specific sperm, a random sperm, fertilizing a specific yet random egg. All right. So, talk about independent assortment. Okay, that produces the initial amount of genetic variation here. Okay, you throw in crossing over, that amplifies it a lot. Independent assortment, random fertilization throws in even more variation, so we end up with enormous amounts of variability here total. Now, how did I come up with this 8.4 million number? How do you calculate the amount of variation or the amount of lineup combinations just from independent assortment? Well, it's simple. Okay, it's simply 2 to the nth power. So 2 to the nth, where n equals the haploid number. So in humans, our haploid number is 23. So 2 to the power of 23 equals 8.4 million. Imagine if we had a species that had 100 chromosomes, right? As the haploid number, 2 to the power of 100. That would tell you how many lineup combinations are possible. Okay, that's a lot of variation. Let's take a look. So this is independent assortment. So we have just a simple case here where we have 2 chromosome pairs. So we see number 1 and number 2, and this is one possible lineup, right? And let's say metaphase 1 of meiosis 1. Both the fathers on the left, both the mothers on the right. Here's a second possible lineup. There are no other combinations, are there? Nope, just those 2. All right, if this is the lineup, and again, they're not showing crossing over here, okay, just independent assortment, you would get this cell at the end of meiosis 1, that one at the end of meiosis 1, then also these 2 cells. So 1, 2, 3, 4 possibilities here, all right? Now, this cell goes through meiosis 2, produces these 2 cells that could become 2 sperm, these 2 cells that could become 2 sperm, or 1 egg, right? These, because they lined up differently, and back in the original parent cell, produces different combinations in what would become the gametes. So we have combination 1, 2, 3, and 4. Now you add 21 more pairs to this, so you get the human number of 23 pairs. You would have 8.4 million of these combinations. Okay, like I said. All right. Now, that's not even considering crossing over. You throw crossing over into the mix, right? Crossing over produces recombinant chromosomes, which combine DNA inherited from both of our parents. It contributes to genetic variation by combining DNA from 2 parents into a single new recombinant chromosome, one that's a mixture of blue and red in some of the figures we've already seen, okay? And again, in humans, it's an average of 1 to 3 or 2 to 3 crossover events per chromosome. And again, they can't be predicted where they're going to occur or how often they will occur. All right, this is showing you crossing over. So we start off with 2 replicated chromosomes, each one consisting of 2 sister chromatids, okay? During synapsis, they line up, and crossing over begins between the 2 innermost chromatids. This is an electron microscope view of a chiasmata, actually 2 of them, the crossover points, all right? Once they separate, now you can see we have 1, 2, 3, 4 different chromatids. We have 2, this one on the outermost side of the fathers, this one on the outermost side of the mothers. They're non-recombinant. They're just like they were before this. The 2 on the inside that participate in crossing over are now recombinant, mixture of blue and red, mixture of maternal and paternal DNA. Now, at anaphase, they separate from each other, as you see here. end up in 4 separate cells, and now we have 1, 2, 3, 4, let's say, sperm cells that are all genetically distinct from each other, right? Now again, egg production, maybe only that one becomes the egg, and these other 4 become the polar bodies that give it the chromosomes, but still, we get recombinant chromosomes and also non-recombinant chromosomes. All right, so let's talk about some numbers here. So random fertilization, okay, we throw that into the mix. This adds to genetic variation because any sperm then could fuse with any particular egg, right? The fusion of 2 gametes, each with 8.4 million possible chromosome combinations from independent assortment alone in humans produces a zygote with any of 70 trillion diploid combinations. All right, that number, 70 trillion, is just from independent assortment alone. Independent assortment and random fertilization. So you have a sperm, which is one of 8.4 million possibilities. It fertilizes an egg, an ovum, which is also one of 8.4 million possibilities. 8.4 million, you take 1 in 8.4 million and multiply it by 1 in 8.4 million, and you get 70 trillion. That means that because of independent assortment and random fertilization, each one of us is a 1 in 70 trillion shot. Now, that number, as bewildering and mind-boggling as it is, does not even take into account crossing over, because we can't predict how often or when or where crossover events will occur. You throw crossing over into the mix, and maybe you add 20 or 30 or 100 zeros to that 70 trillion number. I don't know. No one knows. But imagine, this number, 70 trillion, is already far more than the number of people that currently exist in the human population, 7.4 billion. You add crossing over in, you add maybe 100 zeros to it, imagine all that genetic diversity, okay? Just because of these unique things that occur in meiosis that don't happen in mitosis. That means that each zygote has a unique genetic identity. Now, at this point, a lot of times students will ask me, well, what about identical twins? Identical twins are clones of each other, but they are a result of an event that occurs after meiosis. In identical twins, you have a sperm, which is genetically unique, that fertilizes a genetically unique egg, and then as that egg, that zygote, begins to go through the process of mitosis, it literally splits into 2 separate zygotes, which are genetically identical. Each one then develops into a person, or a copy of whatever organism we're talking about. So that happens after meiosis and sperm and egg production. All right, other than that, there are no 2 people on the planet that are exactly the same. All right, so now I have an animation to kind of show you some of this, and that'll be the end then of the chapter on meiosis. All right.
In sexually reproducing organisms, 3 processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
All right, so that wraps it up for chapter 13.